Okay, am I recording it? Actually, I'm not quite sure. Yes, I'm recording.
Yes.
Yes, I am. Okay. Right. So, class. So let's look at this today. Recap. So we know that meiosis is involved. It is called reduction division. Okay, reduction division because during meiosis there are 2 nuclear division, so that we can produce 4 daughter cells, and each of them is haploid. So we need 2 nuclear division. That's why it's called reduction division. So it's divided into meiosis 1 and meiosis 2. So during meiosis 1, the homologous chromosomes will separate, means the papa and mama separate. Okay. And then during meiosis 2, the sister chromatids will separate as well. Right? So during prophase 1, so the chromatin will condense and thicken to form chromosome. So when they form chromosome, then the homologous chromosomes will start to pair up. So when they pair up, they stick together due to protein complex, or they form the bivalent. So after that, the non-sister chromatids of homologous chromosomes will start to cross over. Okay. Now the pairing process is called synapsis. Synapsis. And then, so synapsis is a process of pairing. Then the sister chromatic cross over. This process is called crossing over. And the point where they cross over is called what? Remember, not chiasma. Okay. So after crossing over, then the homologous chromosomes will start to repel each other, but the sister chromatids still stick to each other. So because of the repelling, it will form the ring shape if there are 2 chiasmata. It will form the cross shape if there's 1 chiasma. So then after that, the homologous chromosomes will align at equator, and then they will separate during anaphase 1 and then anaphase 1 and then telophase. Then after that, during prophase 2, the chromosome, so this 1 chromosome will align at equator, and then the sister chromatids will separate. Pay attention to the sister chromatids, both are genetically variable already. Why? This drawing is not correct. This drawing is not correct. Okay. It must be, is it correct? It's correct. Okay. Both sister chromatids are genetically variable because this particular sister chromatid has crossed over with the non-sister chromatid of homologous chromosome and exchanged the arm. So this arm here and this arm here is genetically different. So these sister chromatids are genetically variable also. Okay, during telophase, the sister chromatids will elongate. So they are called chromosome already. The moment the sister chromatids separate, it's called a chromosome. So this diagrammatically showing in the animal cell, animal cell. So this is during interphase, the chromatin are like thread-like, very long. So during prophase, the chromatin condense and thicken to form chromosome, and you can start to see crossing over also occur already because you can see the loop shape. It's a ring shape and cross shape. Okay. Metaphase 1, anaphase 1, telophase 1. And then this is cytokinesis already happened. So, and then prophase happening inside the cell. Okay, and then metaphase 2, anaphase 2, telophase, and then cytokinesis. So at the end of the day, we got 4 daughter cells. Okay, and then the equator for the metaphase 1 and metaphase 2 are 90 degrees to each other. So the cytokinesis will occur at the equator, cut through the equator to divide the cell into 4 equal sizes. Okay, so this one is grasshopper's chromosome. Grasshopper's chromosome during meiosis, showing you this is prophase 1, and this is also prophase 1. This is metaphase 1 when all of them align at equator. This is telophase 1, because you see each of them got 2 sister chromatids, so it's homologous chromosome separate. Then this is prophase 1, and also looks like metaphase 1. Okay, this one you got to know very well. This one is the cell is being observed from the pole. You see not? I look from the top of the pole, I can see that all the chromosomes are aligned in a circle. So from the pole, the equator is the circle when you look from the pole. So this is actually metaphase 2, where all the chromosomes align at equator, right? Then this followed by metaphase, anaphase 2 here, and then that's it. So this table is for you to read on your own, okay? Comparing between mitosis and meiosis, comparing between first meiotic division and second meiotic division, okay? The same as this one. So now we want to look at why is it that the germ cells in the sex organ need to carry out meiosis. So what is the importance of meiosis? Number one is to perform reduction division. That means because we want to produce haploid gametes, so the germ cells have to undergo meiosis in order to produce haploid gametes. So that when the male and female gametes fuse during fertilization, a diploid zygote is restored. So what is so important about this, that so that chromosome number in the species always remains the same, they will not double of every generation. So let's say our number of chromosomes for humans is 46. So when you produce gametes, each gamete got half the chromosome, which is 23, right? And 23. So when they fuse together, we will restore back chromosome number is 46. So for species, chromosome number remain the same. It will not double. Because when you double every generation, that means every next generation will need to produce double number of chromosomes per cell. They need double the amount of nucleotides to make that double amount of chromosome. So they need more nutrients. If they cannot get, they will die. Okay, so therefore we cannot do that. So second reason is because we want to produce genetic variation in the gametes. So you can see that brothers and sisters, even from the same family, from the same parents, they don't look alike. They're not exactly the same because they're genetically variable. Because the gametes from mother and father are both genetically variable because of meiosis. So why must we make everybody different? Why not make all of them the same better, right? Okay. So why is because if you make everyone different, if let's say, for example, recently we've got a COVID. So COVID started to spread throughout the world. Then if everybody is the same, everybody is not resistant, everybody will be wiped off totally. They die. Okay? But because of genetic variation, there are some people who are non-resistant, some people resistant to the COVID. Then at least some people not resistant die, the people resistant survive. So therefore, human continue to survive on Earth. So it's for survival of the species, not individual, survival of the species on Earth, like what? Like cockroach. Okay? Hang on. They teach very slow. Okay. So how to produce genetic variation? So here we want to see the importance of meiosis. Number one, to perform reduction division to produce haploid gametes. Number two, to produce genetic variation of gametes. So how is it meiosis can produce genetic variation? 3 marks. Number one, crossing over between non-sister chromatids of homologous chromosome. This whole thing you need to memorize during prophase 1. So this whole sentence you need to memorize. Okay. So and then number two, independent assortment of homologous chromosomes during metaphase 1 and sister chromatids during metaphase 2. Number three, separation of homologous chromosomes during anaphase 1. So all these three processes during meiosis make the gametes becomes genetically variable. So we're going to study this one by one. Okay. So number one, crossing over between non-sister chromatids of homologous chromosomes. So what happened? You see here in this diagram, this black color one represents paternal chromosome, okay? Paternal chromosome. And then this white color one represents maternal chromosome, maternal chromosome. So we know that during prophase 1, paternal, maternal chromosome, they pair up and then the non-sister chromatids of homologous chromosome cross over. So when they cross over, you see the resultant is the broken arm, okay, from the synapse will have reciprocal exchange. So you see that originally the paternal chromosome got the allele big A, big B, big C. After crossing over, this sister chromatid now has got a small a, big B, big C. So this sister chromatid is already genetically variable from the original paternal chromosome. Okay, same as maternal chromosome, originally small a, small b, small c. After crossing over, this sister chromatid of maternal chromosome has got big A, small b, small c. So both this sister chromatid and these two sister chromatids are genetically variable because of crossing over. So crossing over during prophase 1 leads to swapping of alleles between paternal and maternal chromosome. Now, all these points here is for you to write the answer when they ask you. So how to elaborate if it's 2 marks or 3 marks, okay? So swapping of alleles between paternal and maternal chromosome. So leading to what? New allelic combination or new allele combination. It means this sister chromatid got a new allele combination. Instead of big A, big B, big C, becomes small a, big B, big C. So it's called new allele combination. Within the chromatids, what is allele? Later on, you'll learn what is allele, okay? Allele is one of alternative form of gene coding for contrasting characteristic. This one I'll explain to you in the next topic today. Okay. So for example, since they give examples, so okay, gene. So let's say the big B here, these are the gene coding for eye color. Then eye color got brown color and blue color. So let's say big B code for brown color, small b code for blue color. Both are the same gene because this gene code for eye color, but both code for contrasting characteristic of the same gene. Why contrasting? Because big B code for brown and small b code for blue color. Okay, so another example that you can use in the daily life to understand or to remember this one better is you look at the car. Okay, there are so many cars on the road. And let's talk about a brand of car. Let's say Proton. Okay, Proton. So Proton is a brand. So Proton is like the gene. And there are many alternative form of Proton that is being sold. So the one that I know is Proton Saga, the very first Proton in Malaysia. Okay, Proton Vera, Proton Perdana, Proton Gen 2. And now I got to know what Proton. So Vera, Gen 2, Saga, Perdana, these are the alternative form of the proton. They are all proton, but they're different allele. They're different allele of the same gene. Okay, so that's a good example that you can remember. So next, number 2, how meiosis produce genetic variation among the gametes by independent assortment of homologous chromosomes during metaphase 1. So during metaphase 1, the pair of homologous chromosomes will line up at equator. Now, each pair of them has got 2 ways to line up at equator, right? Okay, say for example, this one here. So this pair can arrange paternal on top, maternal at the bottom, or paternal at the bottom, maternal on top. Okay, this pair here can arrange paternal on top, maternal at the bottom, or maternal on top, paternal at the bottom. So each pair of homologous chromosomes got 2 ways to arrange at equator. Okay, so therefore this is called independent assortment. Assortment means arrangement. Independent arrangement of homologous chromosome. Why independent? Because how each pair arranged at equator is independent of the other pair. So no matter how this pair arranged, this pair has got 2 ways to arrange. This pair has got 2 ways arranged. This pair also got 2 ways arranged. So they're independent of each other when they want to arrange at equator. They don't care how you arrange. I will arrange in my way that I like, randomly. So each pair got 2 ways to arrange. So because of that, once they've arranged at equator, there are many possible combinations of arrangement that they can get. So what are total number of combinations here? You can see that there are altogether 6 possible different combinations, and therefore 6 possible combination of chromosomes they can get in the gametes. So how do we calculate this? You use probability. The first pair got 2 ways to arrange, and the second pair got 2 ways to arrange, and the third pair got 2 ways to arrange. So altogether, there are 8 possible different pattern of arrangement, and therefore it results in 8 possible different type of chromosome combination in the gametes. So this one leads to 8 different gametes that are genetically variable. So 8 gametes that are genetically variable. Okay, so independent assortment of homologous chromosomes during metaphase 1 will give rise to genetic variation among the gametes. And the formula is 2 to the power of total number of pairs of homologous chromosomes. Okay, so here, this here is an example here. Each pair of homologous chromosomes can randomly arrange at equator. So there are 2 ways they can arrange. The arrangement of a pair of homologous chromosomes in the equator is independent of the other pairs. So when the homologous chromosomes separate, the daughter cells receive different combination of paternal maternal chromosome. Therefore, gametes becomes genetically variable. So you see, one pair got 2 ways to arrange at equator. N number of pairs will got 2 power of n ways to arrange. So therefore here, you check whether you need to write into your PowerPoint. 3 pairs, you've got 2 to the power of 3, which is 8 ways to arrange it equator. Okay? So make sure you write into your PowerPoint notes. So next is independent assortment of sister chromatids will also lead to genetic variation. So this one here shows you the pair of sister chromatids. They are different because of synapses has caused them to be genetically variable, giving rise to different allele combination in the sister chromatids. So when the sister chromatids align at equator, okay, every pair of sister chromatids got 2 ways to arrange at equator. Okay. So again, it depending on how many number of chromosomes you have. Here got 3 chromosomes, therefore 3 pair of sister chromatids. So it's 2 power 3 ways of arrangement at the equator. So you get 2 power 3 ways of genetic variation in the gametes. Okay, 2 power 3 gametes that are genetically variable. Okay. So next, even if there's no crossing over, the moment homologous chromosome separates, it will produce genetically variable gametes because paternal and maternal chromosome originally already genetically variable. So the moment they separate during meiosis, the daughter cells will be genetically variable. Daughter cells will be genetically variable. Okay, even though there's no crossing over, it will also be genetically variable. Okay, so when the homologous chromosomes separate, the daughter cells receive different combinations of paternal and maternal chromosome. Therefore, gametes becomes genetically variable. Okay. So let's put everything together. If the question asks you, let me see. Li Kai-chi said cannot catch the values. Again, what value? Every pair got 2 ways to arrange. How many pairs you have? So 2 and 2 and 2. Okay.
Thank you.
Okay, Kiana, if you don't understand, please let me know. If I didn't read the chat because I didn't open the chat, or else you'll block my view. I only got one screen. Okay, so you just unmute yourself and you let me know lah. Okay, so you see, humans are sexually producing organisms. Every individual look different. So we want to know what are the sources that cause genetic variation among all sexually producing individuals. So number 1, of course, meiosis. Just now we already saw. What are the processes in meiosis that lead to genetic variation among the gametes? So we recap here, okay? A, crossing over between non-sister chromatids of homologous chromosome during prophase 1. Okay, lead to genetic variation in the gametes. Independent assortment of homologous chromosomes during metaphase 1 and sister chromatids during metaphase 2. And the separation of homologous chromosomes during anaphase 1. So all these three processes during meiosis can give rise to genetic variable gametes. So when the gametes fuse, the zygote and therefore the organism that's formed will all be genetically variable. So other than meiosis, what else? Number 2, random fusion of male and female gametes. Because mother's ovary can produce many gametes, which is called the eggs. And father's testes is the place where sperms are produced. So father's testes can produce many sperms, the male gametes. So they're all genetically variable. So when the sperm fuses with gametes, it depends on which sperm fuse with which gamete. And they're all random. So whichever fuse, then the offsprings will inherit those genetic material. So your brothers, sisters will look different because it comes from different sperm and different gametes from father and mother. And different sperm and different gametes are already genetically variable because of meiosis. So random fusion of male and female gametes during fertilization give rise to genetic variation among the offsprings of the same father and mother. Okay, then random mating between organisms within a species. Of course, for human is less likely, but for animals, this is normal. That means to say that every male and female in the animal can have many different, they can mate with many different other male and females. Okay, or for human, it's like, okay, one man has got a few wives, or one female got a few boyfriend or husband. This is called random mating. Okay, because every individual gametes is different, genetically variable. So depending on who you mate with, of course, the offspring again is genetically variable. Okay. Then gene mutation in the germ cells. So as the cell carry out meiosis, when the DNA replicate, then wrong bases could be added in and leads to mutation. So the moment germ cells are mutated, when they form the gametes will inherit the mutated DNA, and therefore it will be passed on to the offsprings. Okay, from gametes, it will be passed on to the offsprings. So gene mutation is another source of genetic variation. Okay, so let's look at it. Because they give rise to new alleles or new genes for that population, for that species. Okay. So gene mutation in our somatic cell, body cell will not be passed to offsprings. Only gene mutation in germ cells can pass to offsprings. And gene mutation is usually harmful. So when a gene mutate, is usually harmful or neutral. It's very rarely that it is beneficial for us. Okay, because whatever we have inherited from our parents, right? Why we inherit from them is because they survive. Why they survive? Because those genes code for features or phenotype that allow them to survive very well in the current environment. So when we inherit from them, when the environment is the same, we will survive very well. So the moment mutation happens, it may lead to an allele that give us the phenotype that is not suitable for current environment. So usually it's harmful. Okay, unless suddenly there's environmental changes and that mutated allele can give us certain features that make us stronger, for example, resistant to COVID. Okay. So you see, number 1, number 2, number 3, right, is reshuffling of alleles that is already present in the population. That means the allele is already in the chromosome. What we do is that we just reshuffle them, just like you take a stack of cards. Okay, this stack of cards represent the gametes from father. Another stack of cards represent the gametes from mother. And then you take one card out from this stack, and the other card out from the other stack. So it's randomly picking. So all the alleles, all the cards are already there. What alleles you get, it depends on which card you take. So it's called randomly reshuffling of the existing allele to different gamete and to different offsprings only. So just playing around with the alleles that are already there in the population, in the 2 decks of cards. Okay. Random fusion and random mating is also the same. You imagine one deck of card represent all the male of the same species. Another deck of card represents, one deck of card represents all the gametes produced by all the males in the species. Another deck of card represent all the gametes produced all by all the females. So all alleles are already there in these two decks of card. Okay, random mating happen or random fusion gametes happen. It depends on which card you retrieve from the male population and from the female population. So reshuffling of alleles. But gene mutation is different. Gene mutation will suddenly produce a new allele in the population. It's like now you've got two deck of cards. You go to another deck of card and retrieve one and put into this deck of card. That means you're taking a new allele into this population that's originally not there. And it arises because of mutation. Okay, can you see that? 1, 2, 3 is only reshuffling whatever allele that's in the population. Number 4 is you produce a new allele into the population. So that will increase genetic variation in the population because of one additional allele there. Okay, hang on. Okay, so this is a past year question. Explain what? Explain how meiosis and fertilization can result in genetic variation among the offspring. 7 marks. So this is an essay question. So now there's no more essay, but they can convert essay to a structured question, convert 7 marks to 5 marks. Maximum marks 5 marks for structured question. So because of crossing over at the chiasma between non-sister chromatids of homologous chromosome during prophase 1. Can you see? I asked you to memorize that sentence is 4 marks, 4 marks, that sentence. So resulting in exchange of genetic material, reject genes. Okay, exchange of genetic materials. Exchange of what? The alleles, not the gene. The gene are still the same. You exchange also is the same gene. It codes for eye color. What you exchange is the allele. Okay, because big B and small b are different, but they are the same gene. So you cannot say exchange of genes, you must say exchange of the alleles. Okay, so during exchange, what happened? The linkage group's broken. So the arm will be broken at the synapse. Then there's a reciprocal exchange. Then there's joining back to give rise to new allele combination in the sister chromatids. Okay, then after that, so this crossing over, right? After that, it's like independent assortment of homologous chromosomes during metaphase 1. So they only give mark for homologous chromosome. They don't give mark for sister chromatids because it's a similar answer, a similar understanding. You see that? Independent assortment of during metaphase 1. Detailed independent assortment. That means every pair of homologous chromosome has got 2 ways to arrange at equator. So total number of different ways to arrange is 2 power n, which is n is number of pair of homologous chromosome. Then mutation, random mating, random fusion of gametes. So all these are the methods to produce genetically variation among the offspring. Offspring means the next generation, the children of the father and mother. It's called the offspring. Okay. So you have learned from this this topic, inheritance of information from parents to offsprings, some terms and definition for genetics like chromosome, chromatin, chromatids. Okay. Meiosis, we look at behavior of chromosome, what happened to nuclear envelope, cell surface membrane, this is cytokinesis, centrioles, importance of meiosis to produce genetically variable gametes, sources of genetic variation in sexually producing organism. So this is meiosis, and then random fusion of gametes, random mating, and also mutation, okay, of germ cells, mutation in germ cells only. So now we move on to look at the next topic where I think most of you will be very familiar and will find it quite easy. It's something like revision, but you must use the term, the method, the format of Cambridge A levels to answer those questions. Okay, so we're going to look at genetic diagrams. And before we start genetic diagrams, we have to learn some genetic terms and definitions that we will use in genetic diagrams. Okay. So now let's look at where's my slide? Okay, this one. So this one is what we're going to do today because I have already done this with another class already. So you have to do it together. Okay. So let's do it. So today, we're going to look at some genetic terms and definitions before we go into genetic diagrams. For genetic diagrams, we're going to look at monohybrid crosses. That means when you cross means you make the parents, okay, father and mother together, or the male and female organism together. Monohybrid means inheritance of one characteristic. Hybrid means what? You know, a hybrid car means a car that use battery and petrol, right? Hybrid. So hybrid is a resultant of mixture from two different parents. So monohybrid crosses means when two different parents cross, the inheritance of one characteristic to the offspring, which is the hybrid. Okay, it's called monohybrid cross. So you're going to look at complete dominance, codominance, multiple alleles, sex link, gene interaction, autosomal linkage, and crossing over effect. So for you in your secondary school, I think you've learned number 1, number 2, number 3, probably until number 4. Then 5, 6, 7 will be new for you in A levels. Okay. So we recap first before we move on. So here I'm showing you a pair of homologous chromosomes. Okay. A pair of homologous chromosomes. Why is it a pair? Can you see why is this a pair or not? Let me see. What happened to my student? Can you see why is this a pair or not? Let me see. Pritisha? Yes, Pritisha. Can you see why is this a pair or not?
Is it because it has two different, like the chromosomes?
Definitely the pair of homologous chromosomes will be different, right? One is from where?
One is from the male and one is from the female.
Very good. So one is paternal and one is maternal chromosome, right? Okay, very good.
Yeah.
Very good. Thank you, Pritisha. Sydney, what else? Why are they called homologous chromosome?
They have the same locus.
Very good. So the same locus means the location where you can find the gene. So the same locus have the same gene, right? Let's say if this is the gene coding for eye color, this gene coding for eye color, this gene for insulin, this gene for insulin. Very good. Thank you so much. Kai Chi, what else?
Yes.
What else? Same what?
They have the same.
Same. There are two more things that are same that you can see from the diagram one.
Hold on, teacher. Wait, I think because I can't think of it like that.
Okay, look at it. Where am I pointing to? This one. What is this?
Oh my god.
Same what? Start with C. C.
The
C.
chromatid.
No, when you got problem, you go to the Taylor's College campus. What?
Central.
So what is this?
Central.
Central.
No, central is the pole. This central is P-O-L-E-S. It's actually the pole, the pair of like centros. No, it's not the pole. Central what?
Centromere.
Yes, centromere. Right?
Okay.
Homologous chromosome has the same centromere position, right? It's located here. This one is also located here. Remember I said some of them on top, some on the middle one, right? Okay, so same centromere position. Last one is very obvious. What? Thank you, Kai Chi. Thank you for the trying out. Jovita. One more thing, they are the same. What is it? Very obvious in the diagram one. Nobody mentioned before. Like, Jovi?
Same size.
Very good. Thank you, Jovi. Same size, right? Because they got the same length, remember? Karyotype. Karyotype is you arrange the chromosome from the longest one to the shortest one, and the longest one you number is karyotype number 1. Correct or not? So same length, and because of that, same karyotype number. Okay? So one from mother in the egg and one from father in the sperm, right? So paternal, maternal chromosome. Same size, same karyotype number, same centromere position. Okay? Same gene in the same locus. They will pair up during prophase 1, right? To form the bivalent. Okay? So therefore, our characteristic will always be coded by at least a pair of genes. Because you say eye color, we must have got these 2 genes that code for eye color, isn't it? Okay? One gene will be found in paternal chromosome, one gene will be found in maternal chromosome. Therefore, our characteristic will always be coded by at least a pair of genes and therefore a pair of allele. Okay? So label fully, so please label this into your PowerPoint notes. So these 2 are sister chromatids. This is the centromere. Okay.
Miss Ho,
Yes.
do you mind going back?
Going back. Okay, going back. Yes.
Yeah, because I need to copy some of it.
Oh, okay.
Okay, done. Thank you.
Okay, make sure you copy, yeah. Okay, right. So this is sister chromatids, centromere, chromosomes. So one chromosome. Each chromosome is made up of 2 sister chromatids. Is one chromosome because these 2 sister chromatids is not yet, they are not yet separated. So the whole thing is still considered as one structure only. That's why one chromosome. Okay. So these 2 are homologous chromosomes. So make sure that you know them very well and you use the correct term when you want to describe the correct object. Okay, when to use chromatids, when to use chromosome, when to use homologous chromosomes, when is centromere, when is centriole. Okay. All right, so this one, I want you to cover this part of your homologous chromosomes in your slide, because your slide doesn't have this box. Your slide, your PDF PowerPoint don't have this box, so I want you to cover it off, or else it's not correct already. Or else our discussion will not be correct. Okay, so please cover it off. So now we want to look at this pair of homologous chromosomes, considering this location here only this location here, considering this location here. Okay, for this pair of homologous chromosome, how many locus are there? How many type of gene? So we're going to look at number of type of, locus, gene, allele, okay, homozygous, heterozygous, okay. So this is what we're looking at, a number of type of. So how many number of type of locus do we have in this pair of homologous chromosome? Are they the same locus or not? Clarice?
Yes.
Yes, they are the same locus, right? So how many type of locus do we have here for this pair of homologous chromosome? One type, right? Because same position. So how many type of genes? Thank you, Clarice. How many types of genes do we have here then? Qiao Yi? Are these 2 genes same type or different type of gene? Qiao Yi?
Same.
Qiao Yi disappeared. Hello, Qiao Yi, where are you?
Same.
Xiaoyi? God, she's here. You're unmute already, huh? Okay, so this is same type of gene or different type of gene? Xiaoyi?
Same.
Same type, right? So number of type of gene is 1 or 2?
1.
One type, very good. So here is only one type of gene. Now for allele, can it be the same? It can be different also, right? Okay, so let's say allele. If you say it's one type of allele, it's because these two allele are the same. Big B, they are the same type. Small b is same as small b. So if allele is one type, that means it can be big B or big B, small b. That means these two allele are the same. Okay, if you say two types of allele between this homologous chromosome, means this allele and this allele are different. So considering this pair, there are two different types of allele. So it must be big B, small b. Then you can say two types. If you say considering this pair, there's only one type allele, that means these two allele must be the same, either big B, or small b, small b. Okay. Now, what about the term homozygous and heterozygous? Homo means same, hetero means others. This word literally means others. Others. Okay. So homozygous means these two homologous chromosome has got the same, homo means same. Okay, homo means same. Has got the same allele. So it's homozygous. So if it's same allele, how does it look like? So it's either big B, or small b, small b. So there are two possibilities that they are homozygous. So there are two types of homozygous pair here. Either big B, so it's homozygous, or small b, which is also homozygous. So homozygous, I've got two types. Heterozygous, that means two alleles are different, only one type, only big B, small b. Okay. Have you written down already in your PowerPoint? Yes, huh? Okay, so let's move on. Now here is where you need to come back again and again to learn up the definition of those terms, and then make sure you use the right term when you describe the right thing. Okay, this is where a lot of students, when they write answers in exam, instead of writing allele, they write gene. Instead of writing gene, they write allele. Okay, and then rarely phenotype, genotype is wrong lah, but usually gene and allele is when they don't use the right term in the right context. So you make sure you learn the definition correctly. So let us go through the definition. So let's recap again. What is a gene? Gene is a unit of inheritance. That means one gene code for one gene code for one polypeptide. We inherit the whole gene into us, and that gene will code for the same polypeptide. So we inherit it from our parents. Okay, so it's a unit because it inherits the entire gene as a whole unit. Okay, it is a specific length of a functional DNA. Functional DNA means a DNA that code for something. So if this DNA, okay, so specific length of DNA here will code for a gene. So this one here will code for a gene. So it's a specific length of a functional DNA. And it's made up of specific sequence of nucleotide or bases. So gene, we've got specific base sequences here, right? Which from there, transcription, translation will give rise to a specific polypeptide. Okay. And then the polypeptide can be an enzyme or hormone, okay, that control the phenotype of an individual. So the enzyme will control catalyze a certain reaction, so control certain product to be formed, for example, pigments of the eye. Okay, so therefore you get brown eye or blue eye. Okay, so it's the phenotype. Now, allele is one of a number of alternative forms of a gene. So the gene may have got many different allele, okay, alternative form. So each allele codes for a contrasting characteristic of the gene, such as big B and small b are the alleles of eye color gene. Both of them are big B code for brown eye and small b code for blue eye. So they code for contrasting characteristic of the same gene. So why they are the same gene, but they code for different characteristics? Because each allele has a slight difference in the nucleotide or base sequence. Okay, so therefore they code for different things. Now, locus is the position of a particular gene in a particular chromosome. Phenotype is the observable characteristic of an individual. So exactly what they are, it includes structural, physiological, or biochemical characteristics. So three things here. Phenotype, when you talk about phenotype, we can talk about structure of this organism, like, okay, got 2 ears, 2 eyes, 1 nose, not so hairy, okay, whether the hair is straight, whether the hair is thick, whether the hair is curly. So this is called the structure that we can see. Physiology is how the body function, like how we move. How we move is because we got muscles, we got bones, we got joints. Okay. How do we obtain oxygen? We use the lungs, right? Instead of gills in the fish and wet skin in the amphibians. Okay. Example lah. How do we remove waste? We've got liver, we've got kidney. Okay, so this is called physiology. Biochemical characteristic is all the chemical reactions in our body catalyzed by enzymes. So what do we code for? We code for the enzyme so that we can carry out the biochemical reactions like respiration, aerobic, anaerobic respiration, like formation of urea by ornithine cycle in our body. Okay, and many others. Okay, so all these are considered as phenotype. And phenotype is determined by genotype and environmental factor. This one you'll learn next time. Okay, phenotype is, for example, if you're born with fair skin, very fair skin, that's because of genotype. Okay, inherit the alleles from the parents. And then, but if you every day you go under the sun, okay, and then don't take umbrella, you don't put sunscreen, then you become darker. So your overall phenotype that we can see on your face, it can be your genotype plus the environmental factor around you. And also plus, let's say your parents are very tall, but you don't have enough nutrients when you're growing up. Okay, when you're, let's say, 10 years old until 18 years old, then you will, and also you don't really exercise, then you will not grow to the maximum height that your genotype code for you or code for that individual. Okay, so your final phenotype still depends on the genotype plus your environmental factor. Okay, now genotype. Genotype is a genetic makeup of an organism with respect to, WRT is my short form, with respect to the alleles under consideration. That means to say, your eye color genotype is what? Is it big B, big B, small b, or small b, small b? So this is called the genotype. The genetic makeup of the organism with respect to the alleles under consideration. It's called genotype. Okay. Okay. Now, what about dominant? This is the part that you make sure that you learn it correctly. Dominant refers to the allele, not the phenotype. Okay. So when you got brown eye, I cannot say that brown eye is dominant because phenotype cannot be dominant. Only the allele can be dominant. Okay. So the allele from one parent that influences the appearance of the phenotype, even in the presence of an alternative allele, which is called the recessive allele from the other parent. So it is the allele that is dominant, like big B versus small b. Big B is dominant because when big B and small b are present, only characteristic coded by big B can be seen in the phenotype. So big B is considered as dominant allele. Okay, what about recessive allele? It is the allele again, it's not the phenotype. Phenotype cannot be recessive. It is the allele that is recessive. So the allele which influences the appearance of the phenotype only in the presence of another identical allele. So small b will not be, the characteristic of small b will not be seen in the phenotype unless and until another small b come up together. So the genotype is small b, then only it will show its characteristic in the phenotype, which is blue eye. Okay. Now, homozygous, which is also called pure breeding, that means we are saying that the two allele under consideration are the same. So the diploid condition in which the alleles at a given locus are identical. So big B is called homozygous. Small b is called homozygous. It is also called pure breeding because these two are the same, so it's called pure. These two are the same alleles, so it's called pure breeding. So this father has got big B. This father is pure breeding for brown eye. This mother got small b for genotype. So this mother is pure breeding for eye color, which is blue eye. Heterozygous means the two allele under consideration are different. Okay, so the diploid condition in which the alleles at a given locus are different. So the allele given at the allele at a given locus, that means the two same locus they are different allele, so it's called heterozygous. So the moment we say crossing means mating between the two parents. We cross the parent to get the offspring, means when the parents mate, the gametes will fuse to form offspring, so offsprings will be produced. So that's the meaning of crossing, means mating. Okay, so that's all I've taught this for the previous class last week. Another class, not your class. So I will stop here so that tomorrow today during the lecture, I will continue from this slide. Okay, so I'll see you today at 12 o'clock. Okay. Okay, today 12 o'clock. Okay, see you later.
Thank you.
Okay, thank you everybody. Have a nice day.
Thank you, Miss.
Welcome. Bye-bye.
Thank you, teacher.
Welcome.